Hi everyone. In this explainer, we'll be taking a close look at a study from Ribeiro and colleagues that gets at a core problem in Alzheimer's disease, memory failure. Their work is interesting because it focuses on a really fundamental cellular process, the regulation of mRNA translation, and asks whether it might hold a key to preserving memory. So here's how we'll break it down. We'll start with the essential background science that really frames the central question. After that, we'll examine the results from two different mouse models, an acute one and a chronic one, to see how their hypothesis holds up. And then finally, we'll wrap up by exploring the potential mechanisms at play and what these findings could mean for Alzheimer's research moving forward.
All right, let's begin with the foundational science. To really get what the researchers were trying to do, we first need to understand the link between protein synthesis, memory, and how this whole process seems to get dysregulated in neurodegenerative diseases like Alzheimer's.
At the very heart of this study is mRNA translation. This is the basic cellular process where the genetic code from mRNA is used to build new proteins. In the brain, this isn't just about routine upkeep, it's absolutely critical for forming long-term memories. That process relies on physically strengthening the connections, the synapses between our neurons. Now, here's where the problem in Alzheimer's disease comes in. There's a growing body of evidence that shows in the hippocampus, which is the brain's memory hub, this vital protein synthesis machinery goes awry. This slowdown or attenuation of mRNA translation is now thought to be a major contributor to the memory loss that really defines the disease.
The study zeroes in on two key proteins that act as brakes on this process. The first one is FMRP, or Fragile X messenger ribonucleoprotein. It's a powerful negative regulator, meaning it can halt protein production by blocking both the initial start of translation and its continuation down the mRNA strand.
The second key player is 4EBP2. This is the main translational repressor of its family found in the brain. Its method is a bit different. It binds directly to a critical initiation factor called EIF4E. By doing that, it physically stops the assembly of the larger complex needed to kickstart the whole translation process for a lot of mRNAs. Okay, so this background brings us to the study's central question. If Alzheimer's pathology is essentially putting the brakes on protein synthesis, what would happen if you could just ease up on those brakes? The specific hypothesis the researchers tested was this. By genetically reducing the levels of these repressors, either FMRP or 4EBP2, could they protect against memory deficits, or maybe even reverse them in mouse models of the disease?
So let's turn to the first set of experiments. For this, the team used an acute model to test a prevention strategy. In other words, could reducing these translational brakes protect the brain from the immediate toxic effects of amyloid beta oligomers? The protocol involved three main steps. First, they infused amyloid beta oligomers, those small toxic protein clumps that are considered a key driver in AD, directly into the brains of their mice. Second, they measured the rate of new protein synthesis in hippocampal tissue using a technique called the sunset assay. And third, they assessed hippocampus-dependent memory using a contextual fear conditioning test. The initial molecular results were quite notable. In the normal wild-type mice, the amyloid beta infusion triggered about a 30% drop in protein synthesis. But in the mice that had either reduced FMRP or were haploinsufficient for 4EBP2, meaning they only had one functional copy of that gene, this reduction was completely prevented. Their protein synthesis levels held steady. And what's crucial is that this protection at the molecular level translated directly to behavior. The amyloid beta oligomers blocked the ability of the wild-type mice to form a long-term memory. But the mice with reduced FMRP, they were unaffected. Their memory performance was completely intact. And the same protective effect held up for the second repressor. The mice that were haploinsufficient for 4EBP2 were also shielded from the memory-damaging effects of the amyloid beta. This showed that targeting either of these brake proteins was effective in this acute scenario.
So the main takeaway from this first part of the study is that by genetically easing up on the brakes on protein synthesis, the researchers could prevent the cognitive damage caused by acute exposure to amyloid beta. This is a powerful demonstration of prevention.
But of course, prevention is one thing, rescue is another. The next, and you could argue more difficult question, was whether this same strategy could work in a situation that better mimics the human disease, a chronic model, where the pathology and the memory loss are already well established.
To test this, the researchers used the APP-SWE-PS1-DE9 transgenic mouse model.
and they used aged 12-month-old mice. At this point, these mice have significant amyloid beta plaque buildup in their brains, and they show the corresponding progressive memory impairments that you see in this model. And the behavioral results here were quite compelling. As you would expect, the standard aged APP/PS1 mice showed clear deficits in their long-term fear memory. But the APP/PS1 mice that also had reduced levels of 4EBP2, well, they showed a completely different outcome. Their memory was restored, and they performed at a level that was indistinguishable from healthy age-matched control mice. This is a critical distinction. We're not just talking about preventing a deficit from forming, this is a functional rescue. Even in an aged animal with months of established pathology, just reducing the levels of a single translational repressor, 4EBP2, was enough to bring their long-term memory function back to normal levels. So, with these results in hand, let's unpack the final part of the puzzle. What's the likely mechanism behind this memory rescue, and what are the broader implications for the field? The authors propose that this isn't just about a global, across-the-board increase in all protein synthesis, which could actually have negative effects. Instead, they suggest a more nuanced mechanism, that reducing these repressors selectively de-represses, or releases the brakes on, a specific group of mRNAs that are essential for healthy synapse function. And there's actually evidence from prior work to support this idea of target specificity. For instance, FMRP is known to regulate the translation of the mRNA for the amyloid precursor protein itself. Meanwhile, 4EBP2 has been shown to control the production of critical synaptic building blocks, things like neuroligands and AMPA receptors. This specificity offers a very plausible explanation for how this intervention can have such a targeted, positive effect on cognition. Now, here's an interesting detail. The researchers measured the baseline levels of these repressor proteins in the diseased mice, and they found they were not elevated. This suggests that the Alzheimer's pathology isn't simply causing the brain to produce more of these brake proteins. The dysregulation must be more subtle, maybe affecting the activity of the repressors rather than just their raw quantity. So, to summarize the study's main conclusions. First, reducing the repressors FMRP or 4EBP2 can prevent acute memory deficits from amyloid beta. Second, reducing 4EBP2 can rescue existing memory deficits in a chronic model of the disease. This all supports the broader idea that correcting these underlying defects in protein synthesis can counteract memory impairment. And finally, this work points to the regulation of mRNA translation as a viable target for future Alzheimer's therapies. This research, therefore, doesn't really end with a final answer. Instead, it leaves us with a critical new question. The next major hurdle is going to be identifying the precise collection of mRNAs that are being controlled by FMRP and 4EBP2 in the context of Alzheimer's. Uncovering those specific targets will be the key to moving beyond genetic models and developing therapies that can fine-tune this pathway with the precision needed to safely and effectively treat the disease.